up photosynthesis, we ended with the Calvin cycle. So we underwent the light-dependent reactions. We know a little bit about light and how much light we need and what form of light we need. We absorbed light, we turned that light into chemical energy, and then we went to the Calvin cycle. So the light-dependent reactions produce NADPH, our electron carrier, and ATP, our other form of cellular energy, so the cell can do work. And those were sent to the Calvin cycle, where we fixed or secured carbon into an organic form. And a big part of what's going on here is actually what's required to regenerate RUBP. So the fact that we can't just put 3 CO2s together to form a 3-carbon sugar. We have to add them onto an intermediate. And so a lot of this work is all about regenerating that intermediate. When we look a little closer at this very important enzyme, rubisco, it actually turns out that rubisco is fairly inefficient. And so when we look at rubisco, it is an enzyme. We do see it as an ASE. It's ribulose 1,5-bisphosphate, so that is actually one of the main substrates. To that we're going to add carbon, carboxylase, and that should be the normal action of this enzyme. However, rubisco also has an affinity or an attraction to oxygen. And so it's called rubisco because it can act as a carboxylase enzyme or an oxygenase enzyme. And so we want to look at exactly what that means again. That means that when our carbon dioxide concentration is not very high, so if we have a low carbon dioxide concentration, rubisco will actually add oxygen to RUBP, and we start losing carbon. We actually use ATP for this. So photorespiration does not make ATP. It actually uses ATP right here. And so we're breaking down RUBP. And this happens, remember, when carbon dioxide is low. And this is usually because water levels are low, water concentration is low, and so we've closed the stomata. The water level is low, we close the stomata. If the stomata is closed, there is no gas exchange. And so when we first close the stomata, we just keep using all of the CO2, and the light dependent keeps making oxygen. And so when we first close the stomata, that's going to begin this shift of the gas concentrations. And when oxygen levels are high and CO2 levels are low, photorespiration will occur. And so that will actually backtrack. We will not make ATP, but we'll use some. We don't make any G3P, and we lose some of our carbon that we had created before. So this is pretty inefficient, but it's really only a big problem when the temperature is really high and the water might not be as available. So what this means is that some plants are just going to be limited. This is just, all they got is rubisco, there's no other choice. This is what their genes code for, and some plants are just going to be very limited. Other plants actually have genetic adaptations to reduce photorespiration. So we do have to keep in mind these alternatives are genetic adaptations. They would have happened first randomly. And then organisms that had this random mutation that survived better, reproduced more, and their offspring were more successful. So we want to look at two main types. We've got the C4 cycle, and then we've got the CAM cycle. The Kalman is actually called the C3 cycle because we're going to be making a 3-carbon compound. The C4 cycle is actually going to include an extra enzyme. Both of these include the same enzyme, PET carboxylase. What PET carboxylase is an enzyme that will fix CO2 before rubisco, but then pass to rubisco. And so PET carboxylase is an enzyme, and what we're going to see is 2 different ways of using that enzyme. Some plants, like C4 plants, and these are actually going to be your grasses. These are going to be your corn, things like pineapple, right? It talks about in the book. What happens in C4 is that we physically separate rubisco from the oxygen. So what's going to happen is that these are called bundle sheath cells. These inner cells are called bundle sheath. And they're right here around the vascular bundle. So right here in the middle is going to be the xylem, the tube that moves the water. These cells here are your bundle sheath. Bundle sheath. And so we're going to keep rubisco only in the bundle sheath. So in C4 plants, rubisco is limited what we call spatially. Rubisco is spatially limited. The location of rubisco is different. In normal C3 plants, rubisco is all here in the spongy and the palisade mesophyll. In C4 plants, rubisco is only made in bundle sheath cells. This enzyme is only produced in bundle sheath cells. In the spongy mesophyll, so in the mesophyll cell, we have our PET carboxylase. And what PET carboxylase does is the initial fixation. So how does this help? How does having a second enzyme help? Well, PET carboxylase has no affinity for oxygen. So it will never bind oxygen, it will only bind CO2. So no matter how high our oxygen concentration is in the mesophyll, PET carboxylase will never bind it. If we keep rubisco in these cells that are just a little closer together, not quite near as much of the gas spaces, then we're just going to be reducing the amount of oxygen that rubisco is exposed to. If we reduce the amount of oxygen rubisco is exposed to, then we reduce photorespiration. Another alternative is called the CAM cycle. The CAM cycle is going to be happening temporally different. So temporal change or difference is going to mean time of day. What this is going to mean is that during the night, we're going to open the stomata. During the day, we're going to close the stomata. Makes sense? When it's hotter and we could lose more water, we keep the stomata closed. At night, we open the stomata for gas exchange. And we don't lose as much water because it's cooler at night. But we don't have any light. We can't do light dependent. We can't get ATP and NADPH. How are we supposed to do the Calvin cycle? What does it matter if we can get gas at night? What happens is that PET carboxylase will work at night, fixing the carbon and waiting. So PET carboxylase will fix the carbon and store the carbon until the daytime. So CAM, and I like this AM, it reminds me of time, AM, PM. CAM cycle at night will open the stomata and fix, PET carboxylase will fix a lot of carbon. Then during the day, we'll transfer that carbon to rubisco. So we still need rubisco in both of these. But what's happening is that we're trying to limit the amount of exposure that rubisco has to any oxygen levels. And so those are two adaptations that can help there. And that is the end of photosynthesis. So you really need to go back and review the individual stages, but also the implication of photorespiration.